Hey, thanks for joining me on my ride. This was a ride covering the 6 papers that were left out of Ralph Baric's 2015 Nature Medicine paper. Unfortunately, I had a little technical glitch at the end of the ride, so the video is going to freeze at the end, but I think the discussion is good enough. It's only about 5 minutes at the end that you miss. So the last 2 papers are also very interesting, but just to run down the list, we are talking about papers which show enrichment of coronavirus on a background of mice to create a SARS coronavirus mouse model. But again, this is an example of enriching on a new background, which allows the gain of function for this virus to then become specific to infect mice. There are papers which show the enrichment of a paper showing the enrichment of a civet coronavirus for the civet ACE2 receptor, enriching them in a cell line which expresses the human ACE2 receptor, and then specifically coming up with the finding that you lose the specificity for the previous animal's receptor as you gain the function of having specificity for the new animal's receptor. So while studying zoonosis, essentially they gave us a little piece of the puzzle in the sense of enriching on a cell line that expresses the human ACE2 receptor would be one way to select for a novel coronavirus that could efficiently infect human lungs. The third paper that I covered is a paper which shows that they are already singling in on 2 amino acids which are crucial for the SARS spike protein's affinity for ACE2, and they modify these they modify these amino acids in a couple different virus variants in order to test their sort of role in the functional affinity for the spike protein for the ACE2 receptor. So again, they are studying things that would show us that they understand where they need to target the gain of function research in order to create viruses which would have pandemic potential. The fourth paper that is in there is a paper by Shi Zhengli and her lab showing that they have found SARS-like coronaviruses in bats and then used the HIV pseudovirus system to study the functionality of their different spike proteins. And I think they studied 8 in this paper, but of course there are many more bats than that, many more bat species, and so chances are that this paper is just sort of the tip of the iceberg of the kind of work that would have been done there. The fifth paper is a paper about the use of culture techniques, and specifically a culture which has mature lung epithelial culture which seems to replicate a mature human lung tissue. So a paper like this or techniques like this would be also useful for enriching for coronaviruses that infect human lungs. And then the 6th paper is a paper also by Baric's lab that studies the role of the ORF6, the open reading frame 6 protein, and its ability to interfere with or antagonize the naturally existing mechanisms that try to keep viral genomes and foreign genomes out of the nucleus of any given cell. And so it appears that this protein has a role in determining how easily the virus can evade this natural resistance to being taken over and to hijack the machinery of the cell to make efficiently make copies of itself. So I think that this paper is an example of how they actually understand how to adjust or to modify, or at least what protein that they need to adjust or modify, target mutations to, target their enrichment to, in order to increase the number of viral copies and the viral load during an animal's sickness. And it may even change the extent to which an animal is asymptomatic while it's already shedding virus. You can imagine a scenario where if you can increase the copy number of the virus being produced in the early stages of the infection, then you would also increase the viral shedding before asymptomatic times. And so again, although I'm reading an awful lot into these papers, it's nevertheless quite easy to read a lot into these papers, these 6 papers that are conspicuously or curiously left out of the current version of the 2015 paper. So if you liked it, watch the ride that comes after this. or rather, if you like this discussion, watch the ride that comes after this. I do think it's a very useful discussion. I go into a little bit more detail and in a conversational sense of how these papers might be related to one another. And again, as I said, I'm sorry for the glitch. There's a glitch in the video at the end. About 3/4 of the way through the ride, the video freezes because my battery starts to die. I'm not sure what I did there. But anyway, I still think the discussion is strong, and that's why I summarized it extra well in the beginning here. So again, if you liked it, if you like this discussion, watch the ride and like and subscribe, and thanks for being here with JC on a Bike. Good evening, Pittsburgh and universe. This is JC on a Bike. My name is JC, and this is Journal Club on a Bike. I'm going to revisit the 2015 SARS virus paper in Nature Medicine by the Baric Lab that is at the center of this gain-of-function debate with regards to the origin story of SARS-CoV-2. The origin story of SARS-CoV-2 was very quickly asserted by the authorities in China, by the World Health Organization, and later repeated by American authorities that the origin of this virus was zoonotic and that it emerged from a seafood market in Hunan, in the Hunan province of Wuhan or something like this. I don't know exactly anymore which names are for which, but I know that it was in Wuhan. And so the idea is, where did this virus come from? That's the debate that's happening now. You see more and more people on the network news talking about the possibility that this virus originated in the lab in Wuhan. And so you know that I've covered the proximal origin of the SARS-CoV-2 virus, the Nature paper, which tried to sort of remove all doubt about the laboratory origin story and tried to forward as much of the market origin, zoonotic origin story as possible. But as I showed you in that video, and I didn't even do it very well in that video because since then I've discovered many more highly relevant papers to sort of debunking the idea that we could have never made this virus, because a number of viral researchers, selected viral researchers, have come out in the news and had their presence amplified in social media. One that sticks out to me in particular is Peter Daszak, as a person interested in how these viruses work and how to harness their biology for vaccine purposes. And that's what Peter would argue that his friend Ralph Baric is doing in this paper. He's evaluating SARS-like coronaviruses and their ability to use parts of them to create a SARS vaccine. That's what they would argue. That's what the methodologies of this paper are supposed to be used for. That's the argument there. Can we make a SARS vaccine from a bat virus spike protein attached to a SARS backbone? That's that paper. So interestingly enough, one of my viewers, a retired English teacher in New Zealand, found a older version, probably the submitted manuscript of this paper from 2015, archived on the internet. And the submitted archived paper has a total of 32 references. I think 5 of those references are repeated. So actually it has a total of 27 references that are unique. And in the final manuscript that's currently available for download on Nature Medicine's website, there are only 23. And in fact, this astute reader also noticed that of the 23 included citations, 2 of those citations did not appear in the previous manuscript and replaced two other papers in that bibliography. So I preface this with bibliographic discrepancies. It was a preview video. And so this video is really for that. This ride is going to cover that particular issue. And now if you will recall, that Nature Medicine paper in 2015, that paper has a chimeric virus in it. And chimeric virus is probably the best way to describe the current SARS-CoV-2 virus. It seems to have a pretty amazing backbone that's very close to SARS, but it seems to have a pangolin spike protein. But very interestingly, 2 of the proteins that are encoded are exactly the same as SARS. And that in general, if you looked at nextstrain.org and looked at how the current coronavirus is mutating as it moves through hosts, molecular biologists have pointed out that to have an exact copy of 2 proteins from a non-related virus would be very difficult to get without mutations, even just point mutations, some variation. To have an exact copy is a pretty strong circumstantial evidential point to make the argument that recombination could also explain this virus. And so there's been plenty of pushback against this, of course, because the idea is that none of these sequences are sequences that we would have chosen, and so it has to be a naturally occurring virus. And here's where the bibliographic discrepancies really are scary, and I'll just let you judge for yourself. But what kinds of papers did they remove from this bibliography? What kind of papers are missing here? Okay, well, I'll just go down the list and we'll see what kind of papers there are. So the first paper that gets left out is titled a mouse-adapted SARS coronavirus, something in mice. So in that paper, what they do is they take a SARS virus and they try to passage it through mouse cells and show that it can adapt to mice and cause virulence in mice, or adapt and develop virulence in mice. So this is actually an example of using passage to create specificity from a virus that's not specific for that animal. It's not in ferrets, which is what you would have had to do to enrich for human ACE2 receptors, but it's in mice, and it shows the principle works. And they're going to argue, the argument that they'll make for this paper is that they were developing a mouse line that they could study SARS with. That's the argument that they're going to make about this paper. But what this paper shows you is that they can passage a virus through mice and through mouse cell lines. That's the more specific thing. They actually pass it through mouse cell lines expressing a specific receptor to enrich for that receptor And then they show that they can generate a SARS virus that can infect mice using the selection process in a cell line that has mouse receptors in it. Ta-da! So that first paper is already a very good example of gain of function, in the sense of using a virus that wasn't good for mice and passing it through a cell line to make it good for mice. And it worked. That's the first paper they took out of the bibliography. You see where we're going with this, right? Every one of these papers is going to be a damning methodological paper, which provides a little piece of the puzzle, another example of their knowledge, the depth of their knowledge when it comes to novel coronaviruses. And that's key. You gotta move over. The next paper is called "The sort of zoonotic pathways are revealed through synthetic coronavirus something or other." In this paper, what they do is they take a civet SARS protein, or a civet, they get a civet specific coronavirus, and they enrich it in human cells, or cells expressing the human ACE2 receptor, and then they show that when the receptor modifies to better infect the human cells, or the cells that are expressing the human receptor, then they lose their ability to infect the civet cells. And so, again, this is a paper that has a sort of biological goal of trying to figure out what changes are important for zoonosis. And here they make some viruses from scratch, and then they figure out how to enrich them to get them to change their receptor affinity from civet to human. And so again, what you're looking at and what you're seeing is the use of biological populations of animals, or cells that express specific receptors. These two methodologies can be used or employed to evolve a virus, essentially to select for interesting viruses that pass your criteria tests for virulence, or for infection rate, or for aerosolage, or whatever. So the next paper is called "The Identification of Two Critical Amino Acids in the Spike Protein of SARS." In this paper, they actually identified two very specific amino acids, God, so terrible driving, that play a key role in the spike protein's affinity for ACE2. And what do? They mutate these single amino acids to observe the change in affinity. So here they are, fooling around with the spike protein to see if they can change the affinity. So again, we might not have come up with the sequence with our computer models, but we could have come up with the novel sequence with this by just mutating. and checking and mutating and checking. Once we identified the two amino acids that were critical for the affinity, we don't need to come up with a whole new sequence, right? Just need to come up with the two amino acids that sit in that position that give us the affinity we want. And so again, you're just hearing how disingenuous everybody is when it comes to talking about what we could or couldn't have done. Because we're still not to 2010 yet, and we're still talking about gain-of-function research. Figure 4 is called difference in receptor usage between SARS coronavirus and SARS-like coronaviruses originating in bats. This paper is done by the now infamous Shi Zhengli, the Batwoman from Wuhan. And in this paper, again, they are making selection based on the spike protein. And in this paper, I imagine this will creep people out, but they use human immunodeficiency virus, pseudovirus constructs to look at the function of different spike proteins and to show that the spike proteins of these bats are already pretty good. Again, we are looking at how spike proteins function and how they mediate cell entry for the virus. And we've been looking for years. And we've taken these papers out of this bibliography because they constitute part of the, the circumstantial evidence, which really suggests that it's at least as likely to be a laboratory virus as to be some as yet undetermined natural virus. So the fifth paper they took out is titled something like well-differentiated cultures of human epithelial cells. This paper describes the ideal culture system on the background of which to enrich a coronavirus to infect human lungs. Sketchy paper to take out of your bibliography. And the sixth paper looks at how the open reading frame 6 protein interferes with the mechanisms that are sort of designed to protect the nucleus from invasion in a cell. And it looks like the protein encoded by ORF6 is interfering with the cell's ability to, I guess, fight off viral genomes, really. We have several mechanisms that are designed to kind of keep foreign RNA and DNA out of the nucleus. And it appears that the virus tries to antagonize this process, and that's what allows it to efficiently infect cells. So this is just another paper showing the depth and breadth to which Baric's lab and Shisheng Li and her work colleagues have dissected the part-by-part functionality of coronavirus in a way that they would rather not have you know about. And they took about 5 or 6 references out of this one, 6 I think, and added 2 more that were both about vaccines. One's even a textbook. So I don't know if this is defensive, like we don't need to help the bad guys, but it seems to me the bad guys are already out there. And the papers are already out there. It seems a much simpler thing to remove these papers from the bibliography to make it less obvious the ease with which this could have originated in the laboratory. And how long we've been working on it. So long